Okay, first of all, everyone, thank you for joining. So today we are conducting the Zoom meeting for presentation for our assignment 1, Microbiology 3. So my group members, myself, Deviani, can call me Deva. So I have with me my group members, Muhammad Yusuf and Firavina Kandit. Okay, so first of all, Yusuf will explain about his point now. I share screen now. Okay. Can you guys see the screen?
Yeah.
Okay. Diba ni change the slide. First is urine.
Yeah.
Ni slide ke berapa ni?
Sorry laptop saya hang sikit.
Tak apa.
Okay, Yusuf, you can start now.
Alright. So today I want to share how what sample collected in microbiology. We have blood, urine, CSF, and sputum. Okay. Now I will explain about how to handle how staff or medical or nurse will take the patient blood and then will clean the skin and insert a thin needle into the vein to draw patient blood and then will to be repeated using other vein to get the most accurate result. Then the blood will be sent inside the laboratory for processing. Okay guys, now we can move the next. This is urine. Okay, for the urine, urine container should be clean and free of the particles or interfering substance. And then the collection container should have secure lid and be leak resistant. If leak resistant container reduce specimen loss and healthcare work exposure to specimen will also protecting the specimen from contaminants. Okay, and then it's good practice to use container that are made of break resistant plastic, which after safe than glass. And then the container. Okay, for the urine, recommended to use primary collection container that hold at least 50 ml, has a wide base on opening of at least 4 cm and wide base prevents spillage, and 4 cm opening is an adequate target for urine collection. Then 24-hour container should be up 3 liter. Okay, for the urine, recommended sterile collection container for microbiology specimen. The container should be have secure close to prevent specimen loss and to protect the specimen from container. Compatible with automated system and instrument used the lab. Okay, collection container should be compatible with the pneumatic tube system if one use urine specimen transport the facility. A leak of proof device in the situation is critical. Okay, now we can move the sputum sample. Okay, this is sputum. Sputum specimen should be collected in the early morning. If possible, collected 3 sputum specimen on the consecutive day unless otherwise instructed. Okay, for the specimen should be kept in the refrigerator until they are submitted into the laboratory. Okay, for the last one is CSF. Okay, for the CFS, to handle needle in the portion, the CSF, the pressure is a measure and sample 1 until 2 until 10 millimeter, and CSF is collected into the vial. the needle is removed, the area is clean, and bandage the place over the needle site. And then you may be asked to remain lying down a short time after the test. Okay, for this, how to collect it and then the sample about the collected in microbiology tree. Okay guys, boleh next. Siapa punya?
Sorry. Okay, so my part, I will be explaining about types of microbes that can be isolated in clinical specimen. So if we take a look, most of the microbes are from the gram-negative bacteria. So first we have the Pseudomonas aeruginosa. So Pseudomonas is actually a aerobic and motile gram-negative rod bacteria worldwide. So if we use gram staining, this is the observation that we can see under the microscope, which is gram negative. Stain dia warna pink, it's pink color, and the shape is bacilli. Bacilli meaning it appears in rod shape. So this one is a closer look of Pseudomonas. Next, we have the E. coli or Escherichia coli. It is actually a common microbe that lives in large intestine of human. Usually, the common disease that E. coli can cause in gastrointestinal tract, which it will infect the colon. Other than that, E. coli is a facultative gram-negative bacteria in human fecal flora. So, as I said, it inhibits in the colon, but some strains of this bacteria can cause disease inside and outside of the gastrointestinal tract of human. So, the picture on the left is a E. coli gram stain under microscope. Shape dia same, it's same rod shape bacilli, and this is a closer look of E. coli. It's blue color actually. Okay, next we have two types of microbe that is from the family of Enterobacteriaceae family. Okay, first we have the Enterobacter aerogenus. Aerogenus ni dia, it's a gram-negative bacteria, and it's a spore-forming bacteria that live as facultative anaerobic. This Enterobacter can be divided into two main species that are Enterobacter aerogenus, this one, and one more is Enterobacter cloacae, but that one is not here. Okay, so the picture on the left, observation under microscope. When we do culturing, this picture on the right, the plate, that's the culturing of Enterobacter aerogenus. Okay, next is Proteus vulgaris and Proteus paninari, which is the microbe that's from Proteus family of Enterobacteriaceae. Okay, so Proteus vulgaris. So mainly focus on another member of Enterobacter family is the Proteus, which is a gram-negative bacteria. Commonly causes nosocomial infections of urinary tract. Usually, these both bacteria won't cause infections in gastrointestinal tract, but in urinary tract (UT). So actually, we have four types of protease, but I only showed you guys two, which is Proteus vulgaris and Proteus paninari. This is observation under microscope. Gram-negative, it's pink and appears in rod shape bacilli. And the right side is Proteus paninari. It shows the culturing of the microbe in a plate. So that's all from my part. Thank you. Proceed to Vina.
They will let me screen share. Okay, you all can see the screen.
Yes, can continue.
Okay, let me continue. Following by that, now I'm going to explain on the infection that caused by the isolated microbes. So previously, Devyani has explained what are the isolated microbes. So now I'm going to explain the infections. So firstly, let us know what is infection. Infection is the invention and growth of germs in the body. So the germs may be bacteria, viruses, yeast, and also fungi. And then the infection can begin anywhere in the body and spread all through it. So firstly, let us see what Pseudomonas aeruginosa can cause. So it can cause urinary tract infection, respiratory tract infection, dermatitis, and also bacteremia. So following by that, E. coli can cause pneumonia, diarrhea, nausea, and also intestinal infection. The third one, Enterobacter aerogenes can cause adult meningitis, skin and soft infection, and also endocarditis. The fourth one, Enterobacter cloacae, can cause intra-abdominal infection, CNS infection, and also ophthalmic infection. The fifth one, Proteus myxophensis, can cause urinary tract infection, which is the main species that cause infection in Proteus species. So the sixth one is Proteus mirabilis, which can cause ascites, one infection, and also chronic suppurative. Then the last one, Proteus venerea and Proteus vulgaris, can cause burn infection and also bloodstream infection. Okay, that's all for the infection.
Okay, Firavina done. Okay, Firavina done, right? Can you guys hear me?
Jadi bah, dengar kita tak? Okay, cuba cakap.
Okay. So we have done with our presentation. So thank you all for joining us. Okay. So we have Amira, Nafila, Amira, Natasha, Ridwan, and Rushafika Diana. Okay, thank you guys for joining. So I will end this session now. Thank you very much.
I'm going to present about possible diagnosis conducted on blood culture and sensitivity, urine culture, throat swab, sputum culture and sensitivity. First of all, I'm going to explain about the blood culture. Blood culture testing is used to identify blood infection that can lead to sepsis, a serious and life-threatening complication. Second is urine culture. This test can find and identify the germs that cause urinary tract infection. Third is throat swab, culture and sensitivity. Throat swab culture is a test commonly used to diagnose bacterial infections in the throat. This infection can include strep throat, pneumonia, tonsillitis, whooping cough, and meningitis. The last one is sputum culture and sensitivity. A sputum culture test can diagnose bronchitis, tuberculosis, pneumonia, cystic fibrosis. Thank you.